Hello, this is Dr. Patrick Hu from Moffitt Cancer Center. We're here with Dr. Amod Sarnak, surgeon in the cutaneous oncology department at Moffitt Cancer Center. Amod, how are you doing today?
Doing well. Happy to be here to join you on this fireside chat.
Well, thanks so much for being here. Tell me, where does T-cell therapy stand for melanoma today?
Well, these are exciting times to be in T-cell immunotherapy. And Moffitt was actually the lead pilot flagship center for a registration trial conducted for patients with metastatic melanoma that have progressed upon standard frontline therapy. And I'm pleased to report that not only did Moffitt put on the most patients on this trial, but we reported a 38% response rate, which far exceeded my expectations. And the data is currently under review at the FDA for regulatory approval.
Well, that's very exciting. You know, patients with melanoma increasingly have options with targeted therapy, immunotherapy with immune checkpoint blockade. Tell me, where do you think T-cell therapy falls in for the regimens for melanoma patients?
Yes, well, like I said, this is really an exciting time for those of us who are taking care of patients with advanced melanoma, because from 1976 up until 1998, there really weren't any new treatments for patients with melanoma. And unfortunately, the response rates weren't good for traditional therapy and patients didn't live very long. But since 1998, there's been a series of novel mechanisms of action uncovered that allow the clinician multiple options in our armamentarium to treat patients. And as you mentioned, that include targeted inhibitor therapy like BRAF/MEK inhibitor therapy for patients whose tumors are mutated with the BRAF gene. And in addition to that, there's been immune checkpoint blockade, IV infusions of drugs that can rev up the body's immune system to attack the cancer. And while these are great therapies, you know, unfortunately, probably about half the patients have disease that ultimately become resistant. And as of right now, T-cell therapy is going to be used or is in development to be used to rescue patients whose disease is refractory to these current frontline therapy options. Probably about half the patients will need some sort of salvage therapy.
Yeah, well, you're also a surgeon, Dr. Sarnak. Tell us about the process of getting TIL cells, because it really is a combination of surgery and medical oncology, right?
Well, I think that this plays to one of the strengths of Moffitt is that you have multiple disciplines, both surgery, medicine, and immunology. They all have to work together in concert. It's almost like assembling a symphony. And if the wind instruments are kind of out of tune, then the whole thing does not sound very good. So as a surgeon, you know, we are responsible for securing the tumor material. These are the tumors that we take out of patients that are sent to the immunology lab for the immune cells to grow. So it's really important to have a surgeon who understands, well, which tumors are able to be reasonably removed and the patients recover in time to get the treatment. And also looking at the scans in order to determine, like, hey, is this tumor going to yield high quality, or is this tumor going to be all necrotic, meaning broken down tumor cells with blood and not really yield a TIL product? And Moffitt was very smart. They involved all three disciplines, both surgery, medicine, and immunology, right from the very inception when we tried to develop this treatment at our cancer center. And I think that's what led Moffitt to be very successful. And it put us in a great position to be the flagship institution to really anchor the registration trial that has just been completed and under FDA review.
Wonderful. Tell me, what happens after you resect the tumor from the melanoma patient? Where does it go then?
So in our center, what the surgeon does is actually prosects or chops up the tumor right in the OR. So it's fresh out of the patient and then dropped right into a transport media. We have exceptional personnel from Total Cancer Care. They pick up the specimen right away and immediately carry it to our cell therapy facility, where the cell therapy technologist then processes the tumor. And I think this is important because the tumor's not sitting around. It has a very low chance of getting contamination. And I think the surgeon being able to pick and choose the best part of the tumor for TIL propagation is really important to get the maximum quality of resulting TIL.
Well, it's very exciting. I hope this gets FDA approved soon. When do you think that's going to happen?
Well, as you may know, the FDA is a little bit encumbered with various problems that relate to the COVID-19 pandemic, as well as the vaccine issue. So I think that's distracted attention somewhat and probably caused a little bit of a regulatory delay in the review of TIL. But I am happy to report that the FDA does an extremely thorough vetting of each and every product that comes across their desk. And what we're hoping for is a final answer sometime in the spring of 2022. But of course, this is subject to change depending on competing interests that are ongoing with the pandemic, especially.
Well, fantastic. Well, thanks so much for being here today with us, Dr. Amod Sarnak, surgeon and clinical investigator in the Cutaneous Oncology Department at Moffitt Cancer Center.
Thank you for having me today, and have a great day.